Welcome back to MACNA 2021, Supporting an Equitable Trade. Our next speaker is a native Australian. He's presenting all the way from Australia. He is here to tell us about why coral taxonomy is important and who should use it. Russell Kelley works as a marine educator, filmmaker, author, and science communicator with a special interest in coral reefs. I'm glad to have him here at MACNA 2021 and hope to see him more in the future as we continue to educate marine aquarium hobbyists around the world about coral taxonomy. Welcome to MACNA 2021, Russell.
Hi there, and greetings to everybody, and thank you so much for the invitation to speak at this important event. I hope that our journey through the wild world of coral taxonomy will be informative and entertaining, and maybe even illuminating. So let's get started. I just want to say that coral taxonomy is something that has developed a reputation of being a bit of a dark art, and so it has, for some, it has a fearsome reputation, but I think that's somewhat undeserved. I think part of the reason that it has this reputation is because it's not well taught, it's not well communicated, and what I do through BYO guides and the Coral Identification Capacity Building Program, which is a great big long name for something that is just basically workshops, Coral ID workshops that my wife and I run. Basically what we're trying to do is just apply really good comms to the art of Coral ID. And there's no denying that it can be challenging and that there are complicated or difficult areas, but they're difficult because we're sort of exploring and discovering at the very edge of science now. And it's actually quite interesting. And that's what we'll see today, is that Coral ID in some parts is really straightforward, but just not well understood or not well taught. And in other parts, we're actually exploring inner space, so to speak. So let's get on with it, and I hope you enjoy the ride. So let's take a view forward of what we're going to cover in this talk. I'm going to sort of present to you that there's a problem with corals, and the problem is what makes them so exotic and so wonderful, and we'll explore that first. We'll then look into why we need names and why the art of taxonomy, the art and the science of taxonomy, is actually important in the first place. And how those names need to be reviewed or chosen carefully with respect to the different end users of coral taxonomy. Then we'll have an overview of the history of the science and how we got to be where we are today. And as I'm sure most of you know, there's been a bit of a revolution on the scene with the advances in molecular taxonomy. And so our little historical account of coral taxonomy will sort of end with molecular taxonomy, with some plain language examples, and then look at strategies for dealing with coral identification from the point of view of different end users. So what is coral taxonomy, or what is taxonomy? Well, it's the science that deals with the naming, describing, and classifying of organisms. And I don't feel that anyone would find that unusual. But more importantly, it's the science that describes the evolutionary relationships between organisms. And part of the reason taxonomists are so nerdy about things is because what we're really describing is, in effect, the deep truths of the unfolding history of life on Earth. And it's important to get it right. It's not just a name, it's not just sort of a fun thing, it's actually the truth. The the truth of life on Earth. And so taxonomy is deadly serious, I would put it to you, but it's actually a fascinating thing. And if you want to go beyond just having a name for your favorite beastie, what is sort of under the bonnet of all of these things is just this fascinating story of how life, the plasticity of life has changed as it's moved around the planet, and how things that sometimes don't even look like each other are in fact closely related. But moving on, let's start with the problem with corals. Now, some people say there's no problem with corals. They're pretty and we love them and we grow them and we got them in our tanks and we're happy people. But if you are the sort of person who works in the field, it can be pretty exasperating, and particularly in the Indian Pacific Ocean, the Indo Pacific, you have a lot of species and a lot of variability. Now, on screen at the moment is the growth form variability of just one species. This is Isopora prolifera, and basically what you're looking at is the variation from the top left of the smear of a colony that's right in the surf zone on the top of a ocean exposed reef, followed by ridges and then strange distorted branches and weird plates, and eventually as you go down the energy gradient towards deeper water or into the lagoon, you're going over the top of the reef, you end up with columns, and again, if you go deep into shaded environments, the growth forms will change even more. Here we have not so much growth form variation, but the variation that you can see in the corallites. That is the little skeletal homes that the polyp animal makes for itself. And this is the corallite variation in a species called Oxypora lacera. And one of the first things you'll learn if you do my workshop is that color doesn't mean a lot. You don't want to rely on color in most situations when it comes to coral ID. Sometimes you can, but not as a rule, you shouldn't. But yeah, like you've got growth form variation, and then corallite variation, skeletal variation, and it's occurring on many scales across many species. And so you end up, if you're going to sort of get interested in coral ID, you end up with this, in the Indo Pacific at least, with this foundational problem. Well, corals worldwide, if we add all the species together, there's about 800 shallow water species. And if you multiply that by their growth form variation, and then multiply that by the variation in their corallites, it adds up to more than you can keep in your head. And this is the fundamental problem with corals. They are just bewildering sometimes. They're just sort of, you think, I know who you are. Particularly, I deal with corals mainly through scuba and field, though I am increasingly being seduced by the world of the aquarist, where you can you have this fantastic opportunity to have this window into the living animal in your own home. But if you're sort of just out on a reef on a dive and you just think, I know who you are, but you're just a little bit different, or you're a lot different from who I suspect you are. And when they're surrounded by hundreds of other species all doing the same variability, it is bewildering. It really does your head in. So you end up with these sorts of situations where you throw the great carnival of life over the top of the whole thing, and what you have is this the exalted richness of coral reefs, the legendary richness, but underpinning it all is these keystone species, the corals. And when you actually stop and try and separate them all out, it really starts to hurt the brain. So I'm going to talk about that story and how to wrangle it and how to get it under control and put names on things that are appropriate. So the need for names. We, as humans, just like to have monikers for things. We like to have names for things. And we need to have, it lets us have a conversation about stuff in our lives. And when it comes to the marine world, what it does is it lets us have conversation about these aliens from inner space, because a lot of these things are not familiar. And without names, we struggle to even sort of put it in a bucket and say it's a bucket of these things. So, like many people would not know what most of the beasties are in here, but in the general public sense. But these creatures are have characteristics, so that we could just sort of say, well, yes, they're all sea squirts, or yes, they are ascidians, and we can sort of go down that path of using ever more technical names to have a conversation about these things. And that's part of the art of taxonomy, is using the right name for the right conversation. So collectively, this is what I call ocean literacy. It's about being able to have these conversations and being able to call on the right name and exchange information with other humans for the purposes of whether you just think they're beautiful and you love them, whether you think they're weird and they're wonderful like these guys on the screen, or whether that's actually something of commercial or scientific or conservation importance. So choosing the appropriate name that has power and application for the end user. Now, we could, here with these 4 examples, we could use these names, the scientific names, but obviously we're going to leave a lot of people pretty cold with these names. And so what we could do is we could call them by their common name, their group common name. But you instantly sort of see that there are problems with common names in that, if you come from my part of the world, Australia, 3 of those names would be different. Depending whether you ever watched TV in whatever language, flipper may or may not be a name. So you might call it a dolphin or something else. So let's explore the use of names in the coral taxonomy sense, in our mind map of coral taxonomy users. And I would highlight to you here the knowledge chain, which is the top row, and the supply chain, which is the bottom row. And essentially what we're sort of talking about is people who are using names for different purposes in their work and interest lives. And so the coral taxonomist is got a job to do, and they supply knowledge products, for want of a better word, which are used by people in science and government. Likewise, knowledge products, some of those knowledge products might be used or varied into the supply chain. And you end up with situations like in this diagram, where you can see that why would the scientists be interested in the products that the taxonomists are putting out? Well, that's because they're interested in doing science. And why are they doing science? Well, it's usually something to detect change. They want to know if the environment's getting better or recovering or getting worse and deteriorating. And likewise, management want to do things like the government will want to fund research and science, and so they will want a species list perhaps, and they want reports, and so they're going to be calling on scientists and taxonomists to provide them with the appropriate ID tools and knowledge products. Likewise, coral collectors will need ID tools at some level in their training or at some point in their work life, because they need to comply with export laws like CITES. And likewise, when stuff is being imported, there's going to be some quality assurance by the industry before it's sent to you, the punter, or you, the recreational aquarist or the public aquarist who have a reputation to defend, and they want to know also that they're getting what they're paying for. And so everyone has a different interest in names, and so the choice of name means that we need to match that with the end user. And in the case of things like supply chains, you can see here, two supply chains contrasted. The use of highly technical sort of scientific type names and ID tools is probably less appropriate in certain third world circumstances where you've got village collectors, but in a highly regulated fishery where it's a more technical sort of production, you might find that you will be able to impose a higher standard of taxonomy, so to speak, in your regulation and ultimately how that transfers up the supply chain. So if you want to have and impose certain things, then you in different markets or in different territories, then you need to be able to take this into account, that you can't just say, I want this species, using a great big long Latin word. You have to produce knowledge products that translate that information powerfully and accurately to the end user, who might be a collector in a village somewhere in Indonesia. As for me, I've got my paw print there in the middle. I sort of straddle all these things because I'm a communication designer and I'm interested in science communication and capacity building. And so I have a foot in all of these camps. But essentially, I think the problem with corals and coral taxonomy is really it's a communication problem. And so we're going to go on a journey now to see how we got to where we are, and why coral taxonomy sometimes has the bad rap it does. So this is a 3-act story. I promise it'll be interesting. I hope I haven't bored you so far with this setup, but we're going to go on a bit of a whirlwind journey across a few centuries to get to where we are today. So I like to think of it in 3 acts, and the first act is the exploration era, which has in itself has several parts. And there's no precise date because there is no actual records of when corals first arrived from the tropical world into Europe. But when they did arrive, that was as curiosities and geegaws from the tropical world. And people were bringing back many things from these exotic destinations in colonial times. And, they were not primarily for scientific purposes. They were, in many cases, for aesthetic purposes or curiosity purposes. And all of this was pre-scuba. So, while you've got these pictures here of people, collecting, that may have been the truth of the matter, but there were no bubbles being blown. This was all just being done by people walking on reef flats or diving in shallow water. Specimens were collected and transported without regard to their ecology, without regard to any sort of metadata about where they came from. And so quite often specimens that have made it into museums will have some very generic the older ones in particular will have a very generic thing. It'll say Java. It won't say anything about where in Indonesia it actually came from. So and their physical condition is often very fragmentary and independent of any actual useful data at all. And so their scientific value is not great, but in many cases this is a problem because they've actually become part of the scientific story, and so we're stuck with them now, and that's a problem that we're going to have to actually deal with at some point in the future. What do you do with type specimens, as they're known, that are broken, corroded, eroded, and of dubious value at all? But what then happened in many cases where these things were arrived back in Europe is they were then swapped as curiosities. And so they would then go between gentlemen collectors and move around Europe as people built collections. And so it's a really tricky business. The exploration era is followed rapidly by this emergence of the natural sciences. And so you ended up with actual expeditions being dedicated to the tropical world. And everyone probably knows of Darwin's Beagle, not so much a coral reef focused expedition, but there were many of these expeditions that were consigned to the colonies of many different countries. And so specimens were arriving back in Europe increasingly with an interest in the natural history that was that they represented. And as natural science emerged, it became fashionable to give things names. And so huge numbers of species were being described because basically everything was new. It was whatever came back was new and it needed names. And so you ended up with this many naturalists, and they became scientists who basically named much of the fauna and flora of the world just from sight unseen. They'd never actually seen where these things came from. They just arrived and they were given them. Again, data poor, and most of the people who were describing these organisms had never seen a reef, had never seen a coral in water. What they had was a bleached skeleton that had been rolling around in a ship for 6 months. At some point, we end up in the 18th century, and natural science is emerging out of the Dark Ages with things like the theory of evolution, and the birth of true taxonomy follows very quickly, because we started putting names on things, but what was really interesting when you got into this stuff was that it was the relationships between and among these organisms that were being described, and understanding at some deeper level how they were related, how they were connecting, and how this told the unfolding story of life on Earth. And so exciting times, massive numbers of species being described, but they were ecologically naive. They were really not great pieces of science because they were disconnected from the environment, and people were just often describing the same species over and over again that was being collected from different sorts of physical environments. Finally, in the exploration era, we sort of end up arriving in the true 19th, early 20th century, and we have a kind of a real maturing of things. And there were many great scientists of this time. You can see James Dana, an important American geologist, biologist, who again might have actually, I haven't been able to work it out, but he might have actually traveled to a tropical place and seen a coral in real life. But what we had was, despite the fact that there was no scuba, there was a greater interest in real academic study of the things that were being collected. And so the geologists who were also looking at fossils really focused on microstructure, because that was the one thing that they could actually measure. They could actually make thin sections of corals. And so they studied what was inside the skeletons. And they were the people who brought forward the modern scientific language which we use today. It has its origins coming out of the fossil geological literature. And obviously, you would have heard the expression, the present is the key to the past. All geologists who have an interest in the life on Earth all gravitate to the modern to understand the past. And so, it was very natural to sort of be drawn to merge and to fuse biology and geology at this time, because it was the great learning experience for a geologist, was to go and see their favorite fossils in real life, the same groups of organisms. And we ended up with these fantastic sort of publications. These, you can see from the drawings and illustrations here. This is the exquisite stuff which has great scientific merit even today, and is still of great value and in the modern conversation about corals. So we've got the morphological character of the species that are being described is becoming better and tighter. Specimens were then placed in museums as types. Type specimens are like the ultimate reference material. A name is attached to a species, and a type specimen in a museum is registered and lodged so that other taxonomists, remember, no planes, people could travel to that museum and compare what they were studying with the type specimens. And it was a great system. It was more about the understanding of relationships between things. And so things were starting to move forward. The downside was that this was a time when it was okay to be kind of bureaucratic, because the world still was measured in a few books and a few, people on the Earth who were interested in corals, say, for example. And it was all something that you could more or less keep in your head. And so they made lots of rules about how to change names, how to determine what name was the most appropriate name, and how to make comparisons. But all of these rules are now coming back to bite us on the bum, because basically hundreds of years later, taxonomy is mired with all of these really sort of tedious rules that mean you can't go forward. Sometimes you actually have to take it. If you ever wonder why coral names change bizarrely and seemingly randomly, sometimes it's because new information has come to light and taxonomic rules of nomenclature are coming into play, and they're saying, well, this specimen in this obscure place in a museum somewhere in Japan takes precedence over this specimen, which we thought was the original type specimen in France, and therefore that name has precedence. And so from now on, we're going to call X, we're going to call it Y, even though we've all been calling it Y for 150 years. So it's tedious, but it's real. Okay, let's get into the water finally, the scuba era. We're going to start blowing some bubbles. Now, this is a photo I hope you enjoy as much as I do. When people were setting off bombs in the Marshall Islands in the 70s, there was money to study the effects of dropping nukes on little Pacific island atolls. And all of the scientific community duly got together and started studying corals and reefs in a way that had never been possible before. And it was the beginning of the, Cousteau had brought and popularized scuba. And in this photo, you can see up in the top left, Charlie Veron, who many of you will know or maybe even met at MACNA, and one of his co-workers, Michelle Pichon, who still lives in Cairns in Northern Australia, Carden Wallace, the Aquapora queen, and really importantly, this guy at the bottom, John Wells. He's the, if Charlie Veron is the godfather of coral, John Wells is the guy who anointed him the new head of the family. John Wells was a fellow I met when I was a student. He was a gentleman geologist from Cornell, and he wrote the original book on coral taxonomy and published it as a geologist would in the Treatise of Invertebrate Paleontology. It was in black and white in 1956, and here he is passing the baton of knowledge to the new whippersnappers on an atoll in the middle of the Pacific in 1976. In this era, taxonomy was finally informed by the living animal and its ecology. So we're moving on. Morphology and microstructure from the geologists and the biologists' perspective are being unified with the animal's behavior and its ecology. And there was a huge explosion in the interest in reefs and a much more multidisciplinary approach, because ecology was being born at this time. And prior to that, you're a biologist or you're a geologist, but ecology allowed you to mix and match all these different disciplines. And so exciting times, and things really moved fast from here on. And out of this era came a really important concept, the concept of ecomorphs, the recognition that coral species would change with environment. And so here we have a hypothesis, which is that this is Poeciloptera demicornis, the nuggety little coral which on the reef crest, but as you come over the top of the reef crest into the lagoon, it starts to branch. And if you go into dirty water or deep water where the light is low, you get these open branching forms. But it's all clearly one species, we thought. But this was applied, this kind of thinking was applied to all coral species. And it was a revolution because you look at the variability in this, and if you're sort of meeting these characters in far-flung places or far different points around the reef, and anywhere between the Red Sea and Easter Island, it's hard to sometimes not want to call them different things. And this is what the work of this era did. It recognized the sameness of the same species, but in different environments. So species in this era were described and mapped. Type specimens in different museums were revisited, and the comparisons were made between whether, specimens and names that were attached to them, and something called synonymies were determined. That means that they sort of go, look, this is the same beastie. It's been described 4 times. And the oldest name, the original name, the name that should be attached to the type that takes precedence is this one. And so the work of this era was to not just map and understand the ecological variability of all these corals, but was to go back to the museums from the exploration era, visit all the type specimens, work it all out, which were the ones that have all been described many times, and synonymize them, and then give one name, one true name to rule them all. from that came geographic or biogeographic patterns. And then ultimately what really was the great flowering at the end of this thing was, in the 90s and early 2000, we had these great publications. In most of you would be most familiar, I think, with Corals of the World, and Charlie's great opus, which introduced the idea of reticulate evolution for corals and shallow water marine organisms. And Carden Wallace with her Staghorn Corals of the World is another really important contribution. But over on the top right there, you'll see 4 green monographs called the Sclerectinia of Eastern Australia, the Sclerectinia, the hard corals of Eastern Australia. And this is what led to Corals of the World. And the Australian Institute of Marine Science basically grabbed Charlie, who was an insect ecologist in the early 70s, and said, Charlie, go and sort out the corals in the Great Barrier Reef and stick it in a book. And that's where it all started. Now, we end up with these monolithic resources, and Charlie had a great flair for A, finding money to publish stuff like this, and B, just the audacity to not just stick to his own little patch and say, I'm going to spend my career studying one group or one family or one genus or one species. He took on the lot. And if that portrait I've described to you of 800 species by all of their variability, if you find that sort of hard to wrap your head around, just think what it took, the audacity to do this, one human. Again, Carden likewise dedicated her life to all of these bewildering Acropora species, and Charlie and Carden worked together on the Sclerectinia of Eastern Australia as well. But what was important is that these things were published. It brought all that information together in one place, and ultimately it allowed us to test the hypotheses, their species hypotheses. And so it was comprehensive accounts, accessible to interested people, and testable for the first time. And so basically the coming of the millennium was the beginning of a new era. And that's Act 3 in our story, the molecular taxonomy era. And this is probably, it's a very unkind treatment of things, but basically it's all gone to hell in a handbasket. It's for some people, it's one way of looking at it. It's a total mess. And what we are dealing with now is a serious revision of a lot of this work. But it's unfair, because this is just science doing its thing. It's science being doing what it does, which is critiquing itself and correcting. If we hadn't have had that other era, the ecological era, the scuba era, if we hadn't had these crash through or crash type people who took it on and made these extraordinary publications, it would never have been possible to test them. And the way they've been tested, and admittedly in some cases found a bit wanting, is through molecular taxonomy. So here's a brief overview of the changes, and we're going to have to sort of treat them as plainly as we can, because there's just no escaping some of the ugly terminology associated with this stuff. But I think I'll be able to shield you from the worst of it. Problem with coral taxonomy is that it is complex. It's complex because you've got all of this kind of Latin traditional descriptive stuff and complex rules of nomenclature, combined with what the geologists gave us, which was all the microstructural words and terminology. Now it's combined that with the terminology of the molecular world, and what you have is like 3 layers of really ugly words and horrendograms, as I call them, the diagrams that the molecular people generate. And it is just difficult to understand if you don't do this for a living. You can't just approach it casually. Part of the other problem is it's done by taxonomists, and these people are not in the business of producing digestible or human-friendly systems for communicating what they do. They spend their lives down the rabbit hole doing what they do, and sometimes it's difficult for them to actually find the time or resources to kind of publish the accounts that would allow people to have a learning pathway to go from what you would call beginner interest through to an intermediate level and then to advanced. And so if you don't have someone to mentor you through this stuff, it can be quite difficult. And that's what I mean when I say things are communicated poorly. They're just not user-friendly. And then ultimately what's made worse is they're mired by these rules that I mentioned earlier, the rules of nomenclature. So here we go. Long, long ago in a journal far away, 1996, the evolution of scleractinian corals inferred from molecular systematics. Now, I'm not sure when you first started hearing about molecules and genetics having a role in our medicine or in environmental science. But this is one of the really early papers. And if you just read under the red line there, the take-home message is in 1996, in their abstract, they say families clustered into two major groups that did not correspond or do not correspond to the morphologically based suborders. So basically they were hoping that, oh, we're going to go and grab a bunch of corals, look at them with these new molecular techniques, and we're not going to worry about it at the species level. What we just want to do is, if it's a family tree that you've got, we would expect that at least the trunks of the tree will be the same. And even in the earliest days, in the very first attempts to look at and compare these two fields of science, there was a train crash. So I'm sorry about these diagrams, but there's no other way to present this, but use the color. 7 traditional morphological suborders, and you can count them here 1 through 7. This is from John Wells and Charlie Veron's classic sort of evolutionary tree of corals. 7 major suborders, and then the molecules on this side, they don't get 7 major branches. What they got was 2 major clusters. And so right at the beginning, the 2 trees were different. And this was something that made a lot of people go, oops. But you think about that, 1996, when was the next time any of us heard about molecules and corals? It wasn't until about 2009, 2010. So over a decade went past before it actually bit us. So looking a little more closely at that paper, essentially, if you look at the morphological tree and you assign these shapes for the 7 major suborders and give them those shapes, clearly, if the molecular tree was the same and matching the classical morphological classification, then all of the triangles should all be together with the other triangles. And that's the simplest way to explain it. Just ignore the names and horrendograms. Basically, the triangles aren't together. Oops, we have a problem. More than 10 years goes by before we get this sort of earthquake paper. And this is a scientific, I won't call it a joke. It's not a scientific joke. But it's a measure of the frustration that everyone was feeling when they titled it "Cleaning Up the Big Mess a Day," right? So you have coral families have names that end in "-ide," And big mess a day is the closest you'll ever get to a scientific joke out of a taxonomist. So cleaning up the big mess a day, again, we don't want to try and understand the diagram other than if they took a bunch of corals and they knew what their names were from the traditional morphological classification, then all the Montastrias, underlined in red, should all be together. All of the Fabias, underlined in green, they should all be together if the morphological tree matched the molecular tree. And you can see here, red, yellow, green, everything's all completely scattered. Clearly, we have a really big problem in trying to align these two ways of classifying corals. So a decade goes by. Again, I'm sort of skipping over a lot of stuff here, but you can sort of see this is one summary diagram. And what you'll notice is that we all grew up knowing about Favia and Montastria, and you can see that by 2012, they don't even exist anymore. In particular, you know, from a practical point of view in the Indo Pacific, they don't exist. And new names were being generated, but you can see here Montastria is split into three. But then, you know, you then have, in the case of, say, Phimastria, then it gets split into three, and then some names are being separated and others are coming back together. It's really quite messy. So the changes started changing. And so this is where a lot of people just threw their hands in the air and said, coral taxonomy, not for me. Let me know when there's some sort of solution, and I just haven't got the time to put into trying to follow this, particularly if the changes are going to keep changing. Excuse me. So let's just look briefly, again, don't bog down in the detail, but look briefly at the types of changes. This is what's interesting. Essentially, we have splitting because things are clearly being misclassified from the point of view of the molecules, and they split a genus maybe into two or sometimes three genera, and they take their species with them. And so that's splitting is a typical thing. But why did Favia and Montastria change their names? Well, they were nomenclatural changes. They were because of the rules. So when you start looking at these things and people are going, oh, gee whiz, we've got to make some changes here for molecular or scientific reasons, then the nomenclatural rules will kick in, and suddenly they say, well, actually, the names Favia and Montastria have priority in the Atlantic, and we really shouldn't be using them in the Pacific. So we're going to call them Phimastria and Dipsastria and so on. And so changes for rule-based reasons were also a problem. And then there were the changes of the changes, which was just science doing its thing and doing more work. But the important thing to note is that a whole lot of names didn't change. There are more names that didn't change than there are that did. And so really, the foundation is still there. It's still pretty solid. It's just a bit exasperating when changes keep changing. Another type of change is lumping. And so here we've got splitting happening, but we also have lumping happening. And lumping is kind of perhaps even more frustrating than splitting for some of us, because I'm in the business of trying to teach coral taxonomy or trying to teach coral ID. And we had two genera. Most of you who've been in the game for a while will remember Symphilia and Lobophilia. And Symphilia was easy to understand. It had great big teeth. It had shared walls. And Lobophilia had great big teeth, and both had fleshy polyps, but Lobophilia had separate walls. Clear, even though they look similar from a texture tissue point of view, they were very different organisms, and it was clearly you had to learn your Symphilia species and you had to learn your Lobophilia species. Near the two shall mix, it's easy. But then you can see down the bottom here, our results, looking at this horrendogram down here, we can see where my mouse is wiggling. Our results show neither genetic nor morphological separation amongst Lobophilia, Parascolymia, and Symphilia. Therefore, we want all the Symphilias. They said that Lobophilia was the name that had priority, and so they moved all the Symphilias back into the genus Lobophilia. And so now when I teach Coral ID, I have to teach 20 Lobophilia species as opposed to 10 Symphilias and 10 Lobophilias. It's just not practical in the way it was, especially when they were well understood. But them's the rules. So sad, but true. But there is good news. The taxonomy is testable. So we've moved on from the opinions of gods on Mount Coral Olympus. And, and that's being unkind to Charlie and Carden and people like that. But we've moved on from that. We can now test their species hypotheses. And we're moving towards a more mature appreciation of coral taxonomy, where species are described and understood, not just using morphology and the eye, the human eye, but testing that and combining it with molecules, molecular information, and also ideally reproductive information, reproductive biology. That's what you need to have a really good species portrait. Now, there will be future changes. There's so much work still to be done, but they're going to be tweaks rather than earthquakes. So that's the good news. So more changes coming, but not like these kind of major, disruptions to stuff that you know and love. Previously successful genus level training capacity building tools like the Coral Finder have now been redesigned, work well, and are available. So that's an ad. You're going to see another ad very shortly, but the genus level is pretty stable, and it's good for doing what we've always done with it, which is resource management. And that means we can get back into the business of detecting change and working for scientific and resource management assessments and capacity building and conservation work. And that's really quite important in this time. And training workshops have resumed. I've just finished one at Heron Island, and it was very successful. So here's the ad, return of practical for the use of Coral ID. The new Coral Finder is got twice the visual coverage, but it works just the same way. It works because it's a visual decision tool. What does that mean? Well, visual decision tools minimize text and terminology. They're not driven, they're not a book that you read. They're a tool that you dip into and you interrogate. And so I've created a system that uses only 10 terms. It avoids assumed knowledge by getting rid of most of the ugly terms and most of the ugly concepts. And it throws the problem solving over to the iBrain supercomputer. That's this thing here, which is the most powerful computer on the planet until Google does something that they can put into a pair of glasses. The human brain is it. And so why does it work? Well, from birth, we're sampling the environment every 200 milliseconds. And that means that your mum and dad say, "Oh, look, doggy." And by the time you're 5 years old, you can recognize 50 types of dog. And it doesn't matter that they're all different shapes. Dogness is pre-programmed into you. And humans have this powerful reference library that just goes, oh, well, it doesn't matter that there are 50,000 species of frogs. It's a frog. I don't have a problem with that. And so this is incredibly powerful visual computer and database, but it's heavily biased towards the topside world. And so when you confront it, the same system, with the marine world, where there are these, other than the things that we actually eat, fish and crabs and so on, most of this stuff is just weird. And so the human brain goes, I don't know. And, you know, like here's a sea pen, a peninsula, and it looks a bit like a feather. You know, that's actually the secret, you know, is looking for a shape. And that's what the Coral Finder does, is it classifies things as shapes, and it puts all the same shapes on the same page. So once we train the human brain to know what corals kind of look like, no matter what weird shapes they have, then you can just go to the Coral Finder and you can use it to search for a shape or a texture, and you get sent to a page which has, pardon me, get sent to a page where all of the things that look like it, in this case, here's a column, all the things that look a bit like a column are on the columns page. And well, let's go and look at the columns page. And sure enough, you know, this guy, your target coral, is something that looks like it is over here, and its name's Isopora. And if you're lucky, you know, in the old versions of the Coral Finder, there would be a code here. It says volume 3, page 421 of, you know, corals of the world. Not quite like that anymore, but there are systems now where in with the Coral Finder where you can be forwarded to the most appropriate resources, and I'll show you that in a minute. So the alternative to something like the Coral Finder, the alternative to visual decision tools, is something like Corals of the World. And which volume do I open? Which of the 3 volumes, 9 kilograms of book, thousands of pages, which one do I open and start randomly flicking through? So visual decision tools have a purpose in that they allow you to do a rapid first cut and get people into the right ballpark. Now, you're still going to need formal resources like these, but you know, like there is a real place for tools like the Coral Finder. I could go on, I won't. Here's the results, before and after results for workshops based around 500 people and 24 workshops. You can see score, prior knowledge score was virtually 0, and the end of workshop is 15 out of 15 roughly. So it works. But there is one big problem area, the acroporas. They're a worry. And again, we don't have the time, but you know, here is a paper that came out last year, and I just refer you to the red line here, and molecular relationships, and revealed that species level relationships within the acropora are incongruent with traditional morphological groups. Now, anyone who's studied the acros will know that, you know, there was the Acropora florida group, there was the Acropora tenuis group, and they have a bunch of things that were kind of crudely similar looking in the way that they branched, or the way that the corallites sat on branches and the branches sat in colonies. That isn't being reflected in the molecules. So that's this tree here. The natural groupings doesn't match what they're getting when the morphological groupings do not match the molecular ones. Now let's look at this lower one here. Significantly, over 50% of the specimens cannot be readily assigned to any of the 408 nominal species of Acropora, suggesting the true diversity of this genus has been vastly underestimated in the most recent taxonomic review. So bottom line is there are going to be name changes aplenty in this space, and anyone who, you know, is saying I can do my acros and I'm totally across it is probably not really telling the truth. Here's the reality. This is Tom Bridge from the Queensland Museum who's working on this. Just read the red again. We're getting close to the submission of 4 papers on the Acropora. That's this year. Essentially, the story is that there seems to be far more species with more restricted geographic ranges than we currently think. Now, what that means is things like, you take something like Acropora hyacinthus, somewhere in the world, there will be a specimen that was collected and it was described as Acropora hyacinthus. And that name will be valid for that location or that sort of region where it was collected. But as you travel around the Indo Pacific, there are all these other things that look a bit like Acropora hyacinthus, which we've always lumped together as ecomorphs, even though they're widely separated. Chances are that these are now what they're going to be renamed and given some of these other names that were treated as synonymies when Charlie and Carden looked at all the type specimens, and they lumped things together and say, these are all the same, they're all ecomorphs. This is the name that takes priority. Everything that looks a bit like this is now going to be called Acropora hyacinthus. Truth is that there might be half a dozen closely related looking things around the Indo-Pacific that look a bit like hyacinthus, and many of them have actually been given names in other areas, but they were then synonymized as hyacinthus. They might then regain their regional names. And for this reason, it's possible that there might end up going from 130 or 150 acros to 300 or more. And don't quote me on that, but yeah, it's a world of its own that no one's going to keep in their head. So pretty well all the books are wrong when it comes to acros. The Coral Finder, when it comes to molecular stuff, the Coral Finder is going to be published roughly annually from now on, and it will just update. It's going to be a print-on-demand type tool, and so it will update and track the science. And so you'll have pretty close to real-time record of what the science is saying in the Coral Finder. The Coral Finder will use species names where the taxonomy is stable, and will point to the relevant literature describing the changes. And for many species, it is business as usual, but I won't be using species names where it's woolly. So here's just an example. There's your Coral Finder, and you're identifying the Coral Finder, and you get sent to this page and you go, hang on, that should be Paraclavarina. And you'll see here it says formerly Paraclavarina, reference 22. And in the front of the Coral Finder, you'll see reference 22, Hoang et al., 2000 and whatever, and that's referencing this paper. So there's a feedback loop in the Coral Finder now, so you can actually see and assess for yourself who changed the name and whether you think, if you really want to go down the rabbit hole, you can. But back to the future, what we're living in now is we've gone from the museum era where it was thousands of things scattered, publications all over the world and museum specimens scattered all over the world. And it was centralized in the scuba era into these big centralized resources. But we're now going back to a world of molecular, diffuse and dispersed publications in a broad primary literature in our molecular era. So it's goodbye to the convenience of everything in one book. So we're almost finished. Thank you for hanging in there. Names are changing, and they will impact different people in different ways. And you can imagine all of these different end users are all going to want their resources. But the thing is, please don't cling to books. In many cases, the books were probably wrong anyway, and they're getting wronger by the day. Some conclusions. Why is coral taxonomy important, and why should we use it? Well, firstly, it's not as bad or as hard as it's ever made out to be. That was always a communication problem. And so we've demonstrated with the Coral ID capacity building program that we can train people in a day to be pretty darn good at genus level coral ID, and that they then learn to see the species. It's a communications problem that can be fixed, especially if you engage the iBrain supercomputer and get away from text. Good tools get results. genus level coral ID is stable and useful for science, management, and conservation, and it's the best way to learn your species. Building capacity is really possible, but we're going to need to treat species level ID with caution. It's going to be challenging, particularly with the acros and some other groups. Probably the what used to be the favias, the Dipsastrayas, and the echinophilias and things like that. I think there are a few surprises in there as well. Supporting taxonomy is important. But in some cases, the truth won't just set you free, it will actually confuse you. So be prepared to, swim in some terminology if you're going to really sort of try and pursue this stuff. But eyeballing it actually is not a bad thing, as long as you're consistent. So the non-taxonomist, if you know your genera well, and I've met so many aquarists who have a better eye for coral, the detail of coral, because they spend all day looking at them in their homes and then in their workplaces. Much better idea than your average student would have. And that means, you've got this incredible resource and community of people who actually know this stuff really, really well. But I would just say, don't cling to the old names. If you want to give something an informal name, by all means, that's really valid, because you say, I have no idea what this is scientifically. I would compare it with something I've seen in corals of the world, but I'm calling it X, and I have my reasons. And that's the thing. Use your informal names consistently and have reasons, and communicate those reasons with others and force them to apply that same rigor and consistency, because then you are being a coral taxonomist. You are doing what we need, which is to apply rigor to this business, as opposed to it's kind of green and looks a bit like a Monty, and it's the green monster Monty or whatever it is. Let's go beyond that, because it's the only way we're going to tighten up our supply chains and have better feedback from the people who are in love with corals, which is the large part the aquarist community, and the people who are supplying the industry. Good species ID really should involve multiple lines of evidence, and we know that there's that portfolio approach I mentioned, but we need more education and capacity building for this, and so this is a process that's going to take a while. It's time, if we're going to have a more mature approach to coral identification and coral taxonomy as a group of coral interested people, it's time for a new holistic view. And that is a view of taxonomy, but it's also a view of the organism. Now, again, aquarists are at the absolute forefront of this, because culturing corals in closed systems is all about understanding the role of nutrients getting into and out of corals through the microbial loops and what's in the mucus on the coral, and the viruses and the bacteria and fungi in the system. And that's how, that mixed with, combined with high resolution ICP OES water analyses, is allowing people to grow corals even better than the real thing. People who work with small systems really know their stuff. And so I encourage you to promote the new view of corals as a holobiont or microbiome, but also a new view of taxonomy, which is this portfolio of lines of evidence. So ACRIS have an important role to play in understanding these extraordinary organisms. And I thank you for listening, and I thank you for all of the wonderful love and work and passion that you bring to this industry, to this hobby, to this pursuit. Thank you very much. And hopefully I'll get to have a chat with some of you in the question session, or if not, one day in a workshop. Cheers.
Thank you very much, Russell. It's amazing your breadth of knowledge with regards to coral taxonomy. I just want to remind everyone that they can now go, or if they haven't done so already, go to macna.org/questions, and you can go ahead and leave some questions for Russell. We have the Q&A right after this session. Russell, yeah, I was wondering when you were going to mention the acros, because I saw that paper come by last year now, and it looks like it's largely up for debate issue. But hopefully in the future, your guide can address that. I just want to mention that too. Russell and I will be working in about the next month to figure out if we can go ahead and put some of these workshops together for all of you to be able to attend. So go ahead and make sure to sign up to macna.org and get on our newsletter for that. Thank you once again, Russell.
Go well.